Oh, yeah. Okay, this is, the COBIDAS was started in mid-2014, an OHBM council statement on neuroimaging research, and it's quite interesting from the standpoint that it's the first time really an organization looked at defining best practices and then putting it to the vote to the entire membership, which you can see in last summer, OHBM put a vote of 153 to 6 to approve this document. And this was a joint effort in a number of individuals in which I'm going to present kind of the summary of findings, but it's also, it went out to the community. So the community made comments and the committee changed things based on the OHBM community. But then you ask the question, why create such a document? Well, there's been a number of papers, including the Eklund paper that came up recently. This one was in 2005 by John Ioannidis, who is an epidemiologist and actually work in pediatric population neuroscience. So I interact quite a bit with epidemiologists, but his statement and paper was why most published research findings are false. Some people, when I describe this, will say, of course he's Greek. That's a Greek tragedy. You would expect such a thing. But I think there's truth in what he says, that there's many published research finding. I won't say it's true or false. I think we live more in a world of continuums and grays. And so most published research findings do have bits of truth. But there are, I think, with all the papers in neuroimaging that are coming out, if they were all true, we should be a lot farther along than what we are now. So what is it in his paper that he describes? So I don't have a pointer, but I'm going to point out a couple things. And this is one of his tables where he shows some practical examples of positive predictive value for various combinations of power. So that's 1 over the false negative rate, and R, which is kind of a nebulous variable that he has in his equation for showing the pulse positive predictive value. So R is really, what R is in his studies are how many true findings are there compared to how many findings that are being tested in the literature. So in essence, how many hypotheses are you testing? And of those hypotheses, how many are true compared to how many are false? And then U, which is the third column, is bias. And I'm going to be going into bias more in depth and giving you some real life examples, the good, the bad, and the ugly, some from my own work and work of my group. But just to give you some examples, if you have an adequately powered randomized control trial, this is the top one, with 80% power, the R value, so the number of hypotheses that you're testing is actually 2. One is true, one is false. And there's very little bias. No bias is 0. Then you have a positive predictive value of 0.85. If you go down the list to, let's say, discovery-oriented exploratory research with massive testing, and you have large amounts of bias there, which is 0.8, you're testing multiple hypotheses, and you're underpowered, then your positive predictive value drops way down to 0.001. So with this in mind, and moving forward and looking at bias, the COBIDAS was covering best practices in a number of different areas. And I decided the best way to kind of conceptualize this is to go through each of these and give you essentially the top 10 or 10 practical points for when you're working on papers to try and apply the best standards. But really, the key factor that I think we came up with is just describing what you do and letting, because we can't designate what the best practices are in every area, but what we can do is describe the best practices for reporting those findings, so just being transparent. So here's 10 good tips for good reporting and data sharing that'll cover areas of reproducibility, replication, and how to understand the results generalizing to different populations. So tip 1 is to provide enough information for the reader to understand potential bias in your study. And I think one of the points, really, the key points of Ioannidis, I think it's an excellent paper to read, is how bias can really influence your results. So this is demographics and clinical confounding. And the best time to think about reducing potential bias is during the design of your study. So I'm going to critique some of my own work here. So I want you to look at, this was published in Biological Psychiatry in 2006. And if you look at, if I'm going to ask you to focus on intelligence testing, the lower 3 columns, and look at adolescent patients, these are patients with schizophrenia, versus adolescent control subjects. You can see the full-scale IQ for adolescent patients is 91. Full-scale IQ for the adolescent controls is 114. So this looks to me like there's considerable bias here in comparing apples, potentially apples and oranges. You look at the verbal and performance, you see a huge difference. The normal IQ is 100. But then look at the parental education level and the paternal and the maternal education levels, because this is not so clear-cut, because schizophrenia, as many of you know, is associated with a cognitive decline. So if you start controlling for intelligence, you're controlling also for a part of the illness as well. So they're actually very well matched on paternal and maternal education level. So there's no clear-cut answer here. Often what I do is present both with and without covariating for something like IQ. But these are issues of potential bias. But I think the point here is that the reader can then look and say, oh, this is potentially a bias, or perhaps not, because they're controlled by paternal and maternal IQ. So what different types of bias are there? I showed you a potential example of selection bias, where individuals are more likely to be selected than others, and there's some groups in which it's much more difficult to get good matching control groups. So we were trying to recruit lower SES individuals, and it was very difficult. Reporting bias, certain observations are more commonly reported than others. Analytic bias, I'm going to show you an example of that. Then there's exclusion bias, those in systematic exclusion of certain individuals. Those of us who are involved in longitudinal studies have to deal with attrition bias. Oftentimes the people we most want to study are those that seem to drop out of the studies. Funding bias, studies that show 97% or 95% of the industry-sponsored studies support the drug of the study. And then there's always the career propagation bias, playing with data until you come up with a positive result. So let me ask you a question. So do darker-skinned players receive more red cards? So I live overseas now in Rotterdam, so I've been learning some about soccer. I still have a lot to learn, though. But this seems like a relatively straightforward question, yes or no. But this data was given to 29 different groups, one data set, many analysis, and kind of looking at how different groups. And you can see that, you can see equally likely. There were several studies that showed equally likely, twice as likely. Most of the studies were in between. And the red indicates those that were significant, and the clear were not significant. And there were two groups which, although the error bars were extremely large, had close to 3 times the rates of darker skinned individuals receiving more red cards. So where does this difference come from? And one of the areas mainly where it comes is in which covariates that are chosen to enter into the model. So you can in essence do beautiful data analysis, and then you have to make some decisions on what potentially confounding variables that you want to include in your model and why. So for example, 67% or 62% chose to position of the person, because I think a goalie is much less likely to get a red card than other positions. Height, weight, age, league, country. So different groups chose different covariates, and that changed the results to some extent. What's the true effect is unclear, but this raises the question, what if we were to send our data to several different labs and say, okay, analyze our data and see if you come up with something that's similar. I noticed there's actually one covariate on here that I would have liked to have seen, that I would have added in the model, and that's the percentage of people on your team who have darker skin, because it's not even one of the covariates. So this sense of what we choose as covariates is really important, and it's good to standardize things. So, and this was mentioned earlier by Bob Cox. Think carefully about the assumptions that you're making when designing, analyzing, and reporting the study. So there's a lot of different assumptions that go into the models that we use, the designs of our study, and I'm going to show you an example now of one in which, this is my own work, which is good news and bad news. The good news was trying to challenge one of the assumptions of, one of the standard assumptions. The bad news is I made a bias, analytic bias error that came out, and which is also an important point, is that science is in some sense self-correcting, and that's a good thing. We see that happen. So when I was at Minnesota, I was involved in a review where we were looking at white matter abnormalities in schizophrenia. And at the time, most of the studies, and this is actually white matter abnormalities of using DTI. So all of the red dots reflect different studies, and you can see they're spatially distributed over many different areas of the brain. And many of these studies were voxel-based studies, and so if you have a red dot in one area, you're testing the whole brain. That means other areas have no result, and so you're only looking at the positive findings in these studies. But there's a certain assumption when you're looking at white matter. So what is white matter? Well, white matter is the high-speed communication between different regions. So if you have disruptions in white matter, that'd be like having disruptions in traffic flow between different regions. Sometimes things flow very smoothly, sometimes you have. And when you think about our assumption when we use voxel-based approaches is that the overlapping regions, so if you're overlapping all the voxels and you're doing statistics spatially expecting some spatial overlap in the signals, and that's an assumption that we have. So it's almost like saying, let's take all these beltways of different regions, we're going to overlap them, and we're going to see if the traffic jams are all the same in all those areas. But you could have traffic jams in different areas, but they could have essentially the same effect on the functional activity of getting to work. Let's say that's your, and I chose different cities. I think Minnesota, Rotterdam, London, but in essence, we put everything in standard space. So I said, what if we were to come up with some mechanism to to look at non-spatially specific white matter regions by taking all your areas. A is taking everything in standard space, and then you create a group and standard deviation image. You then Z-transform all those images. You mask them so you get just white matter, and then you look for these based on the overall mean. Are there areas which have these potholes or white matter areas which are go downs that are more spatially individually tailored and spatially specific? And so we did this, and we found between patients and controls there was a significant difference irrespective of your minimal voxel cluster size. But in the analysis, in the design, in the mathematics of this, there was a bias, and that was reported, and I had to deal with with that. So potholes and moleholes bias, and there's been a couple papers that came out, and those have been corrected, and there are other methods used using this kind of same approach, but this analytic bias that can happen, and also the self-correction, but it's also challenging certain assumptions in the literature. And there's other examples of analytic bias that over time they get worked out. So tip number 8 is establish fixed pre-processing and processing pipelines if possible. And going back to the Ioannidis paper, so the greater the flexibility in designs, definitions, outcomes, and analytic models in a scientific field, the less likely the research findings are to be true. And partly because we have this push for positive findings, and there's so many, if you think of, in the earlier talks, all the different possibilities from, I just went backwards. No, all the different possibilities for different analytic processes. So time shifting, motion correction, linear versus nonlinear registrations, differences in brain templates. If you were to go through and calculate the number of different pathways, how many different options that you have, it's huge. And one of these ways is going to give you a positive finding if that's your goal. And so if you can fix pre-processing processing pipelines and then deviate from those with certain justifications, I think it will help this. And then use provenance algorithms to document your data processing. So tip number 7 in the top 10 tips for good reporting. In the statistical models methods, just make clear what your variables are: dependent, independent models, inference testing. Consider analyses that make fewer assumptions about the data while still providing the needed inference, such as perhaps bootstrapping and permutation methods. Provide the rationale for the choice or lack of. There's certain cases where you may not want to do multiple testing, but in most cases it's true. So what are you choosing, and why are you choosing that? And there was a paper in 2012 showing that only 59% of fMRI studies are corrected for multiple testing. I think that's changed. I think, and that's one of the key points of the COBIDAS document is really you need to account for multiple testing and then describe the rationale for that. So if possible, perform a replication in your study, and not everyone can do that, depends on the sample size. I work currently in Rotterdam in the large population-based study of child development, and with large sample size, you can do this within your study. And we have an example of that in which actually for 2016, we have the OHBM award for best paper, which I'll show you here. So what we did is we looked at resting state networks in school-age children. And we did a bootstrapping approach where we extracted we had about 500 samples of resting state networks in children. And we pulled out 50 and we ran ICA, and then we used variation of a dice coefficient to see which networks were there. And then we threw them back in and did it again. And we looked at the stability of resting state networks. And we also looked at, we also presented not only the typical resting state networks, but we also showed noise networks, which are in the lighter gray, and then these are networks we're not sure what they are. There could be noise, when you look at the frequency spectrum versus the spatial characteristics, it's not completely clear. But there's good news that like the auditory, the default mode network, parietal, visual, right, many networks are very robust. Out of almost 100% of the time, 100% or almost 100%, they're always there. There are some networks, like the sensory network, that was much less stable. And there was also noise networks that are highly consistent, usually the flow artifacts, but other ones are there. So, and the data, we actually did, this is work of Ryan Metzel, who actually worked a lot to run all these analyses and get all these done, but these are all freely available to download through Nitric. So this is really actually very important. Provide enough information that your study could be both reproduced and replicated. Reproducibility, and we went through on the COBIDAS document, what is reproducibility, what is replication, what does it mean? I mean, for example, test-retest reliability, there's a nice figure. You could have it done within the same session, or you could have them come back a week later, and what does that mean? But reproducibility, in essence, is an investigator independently obtains the same results with the same data. That's like the soccer example. And replication is a different investigator independently obtains similar results with different data. And people have probably heard about this first came out as the voodoo correlations. But one of the things they brought up in this paper was the high correlations are all the more puzzling because the methods sections rarely contain much detail about how the correlations were obtained. And then more recently, the estimating the reproducibility of psychological science in the open science collaboration, in which I think 39% of papers were reproduced, looking closely at 100 different papers. But they also reported potentially problematic practices include selective reporting, selective analysis, and insufficient specifications of the conditions necessary to obtain the results. And this is, I think, one of the main points of the COBIDAS document is just describe what you do so that other people can understand, they can come up with their own decisions, because we're not going to define we're going to define best practices of reporting, but there's so much variability in how people of the data itself and how you analyze the data. And here's the open science collaboration, the actual data, which I find kind of shocking in some respects, that you take 100 papers and only 39% are replications. So the effect sizes were on the order of half. But this is a graph showing the replication effect size on the y-axis and the original effect size on the x-axis. And then the red, blue is significant and red is non-significant. And you can see the differences. So data sharing. So I think this is a large push, especially I was just at the hackathon, and this is one of the main pushes of open science, the open science SIG. Make your data available for other researchers to use. If it's available, then people can go in and look and rerun the analyses or run different analyses and try to understand more aspects of the results that you do get or don't get. But it's best to think about data sharing before you start your study. I mentioned in a large population-based study in child development, I interact a lot with epidemiologists who are less willing, I think are less willing to share data. It's not as much in the culture as in the neuroimaging or the emerging neuroimaging culture. But it's best to, when you're writing your IRB, when you're writing your medical ethics, put in statements for consent. And I think for the US and Canadian side, this with the NIH funding and requirements of data sharing, this is moving that way. So, and then put funding to support data sharing in your grant. We were part of the MIND Research Network, and we were sharing data, and it's actually quite a bit of work and to get to be involved in data sharing. And I think that's one of the obstacles in data sharing, is that there's so much work. But if you think about data sharing from the perspective of three different groups. You have researchers, you have funding agencies, and you have the public. So if you think about it from the public perspective, as long as you keep things confidential, data is de-identified, there should be, I think, the general consensus is from the public, they're going to be very much in favor of shared data. And funding agencies get more for their money through data sharing, having it accessible, more transparency. It's the researchers, it's us who have the most to either gain or to lose through data sharing. I'll argue that we have much to gain through data sharing. We can address research questions not possible with data from a single lab. If there's some mechanism of credit, like data paper citations, that would be really helpful for data sharing, and recognition from data sharing. But the drawback is you also, and this I hear in the group, my collaborators that I work with, that you spend a lot of time collecting the data, and then you want to publish with the data and have time. So that can be done with like grace periods where you have time to analyze your data before the data is released. And I know that's currently being done with things like the ABCD study. But there's a lot of open data and open platforms and in essence, this fear that if I make my data public, then other people are going to use it and I won't get credit for it. I mean, a lot of the open source data, software, and all that is really driving the field. So I had a few kind of posters, which when I was involved in a data sharing organization in Leiden, that sharing also helps developing countries that might not have the resources to collect the data, but they have networks and they can use their resources to look at data in different ways. So tip number 2 is if your study design and analyses are solid and you obtain a negative result, then publish a negative paper. I know it's hard. Make sure that a power analysis includes. So I was looking at my data, my papers. So how many positive, what's my relationship to positive versus negative findings? And I have a ratio of 9 to 1. So does that mean that 9 of my hypotheses, I'm very good in choosing hypotheses that are correct, or I don't publish negative findings? I'm not sure. But I can say if you're always getting, if you're not publishing any negative findings, then what does that mean? I think and tip number 1, be transparent. No study is perfect. I've shown you my successes and some errors. No study is perfect. Just be transparent in what you do, put it out there, and then the readers can make a decision on different aspects of bias or the findings. Okay, I will thank all the people in funding agencies and students, and then I think probably now. Now I'm done with my talk, but probably we could just take questions as an entire group up here now, or I can take specific questions, but. Yeah, okay. Any specific questions about the COBIDAS document or data sharing or anything in that?
Hi, thank you for those tips. You mentioned briefly that there might be some sort of sourcing the original study, or one of the benefits for the funders is that they then have the research going further. And I was wondering if maybe you could speak a little bit about some ideas of how this would work in terms of the open platform, whether or not you still, if you were to publish someone else's collected data, reference the source that paid for it, and how you might go about referencing that lab that collected it.
So, if I understand the question correctly, is if you share data, and there's groups that share quite. I mean, the Human Connectome Project is sharing. The NYU are sharing. There's the Abide Data Consortium and that. And usually, what they do is write a data paper that is cited. So one way to get credit for that would be to have an S index, a sharing index for different groups, where if you're involved in open access data sharing, there's some way to get credit for that data sharing. That's one point, I think. Does that cover one point? And the other point is that's giving credit to the lab for usually data sharing can be more labs, but the other is I think you should cite where the data came from. If you're trying to replicate a study, just that should go in your paper that you're trying to replicate a study and where the data is coming from. And those are usually linked in different things. And there's been some recent examples of actually papers that are completely open. You can go and access the data. You can run the analyses kind of in open format in order to reproduce the results. I haven't done that. It seems, I'm not actually sure how much effort that took. It'd be an important question to know.
Discussion period.
Okay.
So we stay if you can. So we have a discussion period now, and so I'd like to ask all the speakers from this morning who are here if you could come up. It would be fabulous, and we'd like to have a chance to kind of reflect on the morning and have a broader discussion. So come on up, and this is a good chance to ask any other questions. So don't be shy about asking. Okay, yeah, see if these are on. Yes, maybe. Yeah, we built in time for these discussion periods because I think a lot of times people they listen to the talks, don't necessarily feel like you can sort of explore an issue and raise it. So we kind of built in these times this year to let people do that. So if you have a question or discussion point, come on up. If the panelists also have points then or things to discuss, then you can go for it and raise them.
Room is too small.
The room is too small. How can we get a bigger room?
Yeah. When Tonya was talking, it reminded me of something that's been around in the computing literature for a while, and that's the idea of the self-executing paper. That is, a paper that comes with data and scripts so that you can reproduce its results. That doesn't mean they're right. They may be wrong, or they may be understated, overstated, who knows, but at least you can say, oh, okay, they did this, you can execute it and say 4.7. So, and they got 4.7, so then, now this is a very difficult thing to do, especially in fMRI where there's a lot of data, and then the scripts, it's not just one script that executes all of it, because you have scripts for the pre-processing, and then scripts to do the group analysis, and then probably other scripts to produce figures and so on. And so to do all of that, package all of that up, is a tremendous amount of effort to organize it so that someone else can understand it, because probably you don't really understand all of it at any one point, even if you did all of it, especially, and if it was distributed among a bunch of postdocs and graduate students, then nobody really has got it. But even an attempt to do this is worth something. I think it's a lot better than just sharing the data and say, here, go ahead. Because the script, the data is one thing, and processing it is, you know, just a big thing. And an example that people, a very specific example that we saw in resting state analysis a few years ago is that people do band passing of the data sometimes, and then they filter it in other ways, motion regression or something. And the thing is, they wouldn't specify how they did this. They just say, we did A, then B, then C. But they do them simultaneously or not. But in the case of band passing followed by motion parameter regression, the order in which you do those, 1, then 2, 2, then 1, or 1 and 2 together, will give you different results, seriously different results. And people never specified this. If you had the script, you'd know. But people didn't specify it because they didn't know, which is another thing of getting back to my plea, is someone should understand what you're doing. A little, at least a little, please.
Is there a related comment or question back there?
I walked in late, so I don't know if it's related, but I had a question for Dr. Cox. I was hoping in the description of your talk, it mentioned that you might talk about the challenges that come in pre-processing with short TRs. Do you have anything to say on that?
Yes, but I didn't. There were a lot of things that didn't make it into my talk because of lack of time, both on preparation and lack of time in 30 minutes. But very short TR stuff, as Larry Wald demonstrated, you can clearly see. I mean, if you look at the time series as opposed to the power spectrum, you can very clearly see the heartbeat and respiration. I mean, it just goes up and down and up and down. It's a very dominant feature. If resting state stuff at that like that is, I mean, it's the most obvious thing. If you have a TR of 0.2 seconds, you look at it and you say, what the heck is this stuff that's going up and down real smooth? And you say, wait a minute, that's about a second apart. Then it's obvious. And so, but then if you're going to process that, I mean, that is, the simple models are used for time series statistics, like AR or ARMA models, aren't going to work, because the data, the fluctuations in the data that are of no real interest, the rubbish, the noise, are more complex. And we don't see that with TR as 2 seconds. It's all it's lost there. So there is a simple example of something that can be done, something that has to be done.
I think Gary has maybe a lot of thoughts about this too. You've been writing about this for a while, Gary. Years.
Yeah, a lot of people have been enamored of the better statistics that apparently you get with the short TR acquisitions, and people as Bob just said, often apply the wrong statistics to this. That is, you end up without taking into account the serial temporal correlations, and so the statistics are often inflated, and you think you're getting really awesome Z or T scores when in fact most of those measurements are not independent. And so you have to be really careful when you draw inferences about something with a TR of 0.1 or 0.2 seconds. You're getting hundreds of data points where you used to get tens of data points for the same amount of time. And you just have to be careful that you apply the proper statistics to understanding what those seemingly separate measurements give you. So the early results, and there are lots of classical examples of getting Z-scores or T-scores of 30 or 40 or 50 and saying, aha, I really have improved my statistical inference. You have to be pretty careful about that. With respect to what Bob was just talking about with physiological information, in fact, cardiac and respiratory signals are not very sinusoidal. And so you get second and third and fourth order harmonics that with a TR of 250 or 300 milliseconds, you'll miss. And so if you think you are properly denoising by simply doing a bandpass filtering at, say, 0.2 or 0.3 or whatever hertz, in a very rapid time series, you may actually be aliasing in quite a bit of energy that we're finding from the higher order harmonics in those physiological signals. So you have to be a little careful that you think you've totally removed those signals, but in fact you haven't, even by doing a rapid acquisition and bandpass filtering the first or even the second harmonic. So I think a little caution has to be applied to the very rapid acquisitions.
So in my lab, like many labs, I think now we've put a lot of stock in trying to go fast and we have a TR usually around 400 milliseconds now. And one of the ideas behind that is that it'll reduce, allow you to do more bandpass filtering of noise. And I guess one question is, does that really actually improve group statistics for many people, right? I have people compared. So we did a comparison in 90 people, some of whom have a multi-band and some of them whom don't, different people, right? And it was actually really quite comparable for us at the end of the day in terms of a group statistics. So I guess I'm wondering what other people's experiences are with that.
Can't speak with experience, but that's kind of what you'd expect, that in fact inter-subject variability is generally much larger than intra-subject variability, so that even if you measured your processing was in some sense perfect on each individual subject, it may not actually improve your group statistics that much. Another thing that I've thought about that slipped my mind in my previous ranting was that Gary and I were talking about the noise, but the other side is the signal. The signal model will have to be a little more complicated because right now, with TR, say 2 seconds, whole brain, the signal goes up and the signal goes down. And we use a pretty simple model for that because it goes up to a brief stimulus. We have like 3 data points or 4 data points in the up and down part. You can't fit a complicated signal model with 4 data points. But if you had 40 data points in there, then you may want to fit a more intricate model to a shape model. Otherwise, you'd be leaving signal on the table, so to speak.
And I think with the advent of much more efficient acquisitions and faster TRs, exactly what Bob said is that people are going to find that the HRF is not the same in various parts of the brain, and the ability to look at these rapid acquisitions may give you more information about variability. We've seen, for example, that Just hemodynamic delay can be substantial between various parts of the brain, but the shape will also change as Bandettini and others have shown. So that I think the simple assumptions that people are making now is that the canonical HRF is perfect for the entire brain, is going to soon be found to be not a very good model, and people are going to start looking at much more complicated ways of modeling the data across the brain.
Yeah, maybe I could sort of underscore that point, because I think a number of people over the years have done work on modeling HRF shapes, and there's actually some quite good things you can do. But I think one of the limitations has been that in small samples, power is relatively low. So if you try to model a more complicated shape, then you pay the price in terms of efficiency and power, if the canonical model is okay. But I think what's going to happen now is two things. One is, as people start to analyze larger and larger data sets, the added value of a more precise hemodynamic model is going to go up, because there's going to be lots of power, and we're going to care more about getting it right for every brain region. And then number two, I think one thing that can really sort of come, into vogue really for the first time, I think, in a broad way, is actually modeling latencies and changes in shape, with that additional power. So I think that's a high growth area.
Well, thank you all for the very interesting talks. I had a bit more of a general question. Speaking of these advanced methods and analysis, these sorts of issues, how much do you think this is influenced by the choice of experimental task in terms of the analysis, in terms of the type of analysis you can do, and some of the bad press that MRI research has gotten? How much do you think the actual experiment has to do with that?
Are you asking how or which methods to choose relative to a certain scientific question?
In a way, I guess it's more to do with, we talk a lot about how can we advance the type of analysis we can do, but obviously it's not all suited to the same type of experimental design. A lot of it works when you have checkerboards or a simple visual stimulus, but how do we also cope with more advanced experimental designs, like a complex conditioning paradigm? How do we reconcile using the most advanced analyses with also complex experiments?
Right. Maybe I'll respond in terms of fMRI data acquisition, like in which, when you present the scientific question, you need to ask yourself first, do I want to look at the entire brain? This already imposes some limitations on the spatial resolution that you can get on the TR, etc. Or do I want to focus on a small part of the brain and really image at high resolution and try to get to the level of cortical columns or cortical layers, etc., etc. So that's the very first question that you need to ask yourself. Then in terms of data analysis, I like the approach of looking at various data analysis ways to ask or to sort of try to answer one question. For example, you can do the bread and butter general linear model, or you can do decoding, which shows that you are able to decode various conditions that you may be interested in, which means that your data has the information that allows you to decode it. It's there in the data, in the functional architecture. And maybe Tor can take from here.
could say a drop about experimental design. I think is really critical, and in general, it can have a huge impact. Of course, I'll just say one thing. What I think is one of the sort of most overlooked ideas about how experimental design can impact replicability and reproducibility is even if you have a really good design, an optimized design, let's say. You're still usually in a study only testing sort of one very narrow slice, right? One particular design. And in some studies, even some very large studies, like with the ABCD study, 10,000 people, there was a lot of discussion about whether to use the exact same timing, a sequence of trials where the timing is fixed across every individual in the 10,000 person sample. And while there are arguments why that would be okay, or good, or desirable from an individual difference perspective, in terms of generalizability and reproducibility, that's actually not a good idea, because then what you're imaging with very high power is one exact sequence of trials in one task. And if you change the sequence of trials to another sort of random set of event timing and so on, you're going to get a different answer. How different? We don't know, right? So one of the basic principles, I think, that we'll talk about a little bit this afternoon is if you want to design a study to extract general features and generalize to some task broadly, then you want to build in variation in your design. So if you're studying inhibition, ideally you'd like to build in variation across different kinds of inhibition with different stimuli and different timings and so forth. And that actually gives you a better opportunity to extract something that's generalizable. So that's just one of many kind of aspects.
I'll say something as well. The more complex your paradigm, your analyses are, the more difficult it is to understand or to model, to understand the power. And if power is lower, then it's more likely that you're getting false positives. So I would say the planning ahead, depending on what your specific hypothesis is, and making decisions on the complexity, if you can keep it simple or more straightforward, or building it on more simple models until you reach your complex model, that might be one approach to take.
Okay, so I have to ask this question mainly for Gary, and anyone else wants to comment too, but I found that the potential for deep, non-invasive neurostimulation to be really profound. I'm thinking about the cell paper that you talked about. And if you can put electrodes on somebody's head and create some kind of resonant electrical frequency across the two that lines up and then actually produces deep brain stimulation without going into the skull, this has huge growth potential and implications, right? One really simple question is, I guess, they did that in mice. Do you think you can even do it in humans? Right, that particular thing, right? Because the heads are a lot bigger.
Yeah, it's a cute paper because they did the experiment in rodents, but they show pictures of human brains, and the implication is that all is perfect in human brains. And what we're finding is that, in fact, currents don't actually go in nice little paths between one electrode and another, just as you model with a nice uniform complex conductivity that doesn't have a lot of features in it. But it turns out that it looks like the currents are going along sulci, where there's nice ionic conductivity in the CSF that is much higher than in the tissue. So you don't have a nice banana that connects to electrodes. What you have is a jagged thing that goes along wherever most of the CSF is, and maybe that will make a big difference in terms of whether you can actually get a signal in the deep brain or not in a thing as large as a human head as opposed to a rodent head. So I think the jury's out. I agree with what Tor said, that if this could actually work, where you can electrically stimulate quite deep regions of the brain, that's something that TMS doesn't do very well because the magnetic field broadens out as you go down, and if you can really focus by this temporal interference, especially when it kind of requires a nonlinear interaction in order to see an actual frequency that's present at 10 hertz or 1 hertz or whatever. That's a really interesting concept. So I think this paper will stimulate a lot of interest, and in fact, it stimulated ours a lot. We had already started looking at multi-channel, different frequency and different amplitude modulation of TDCS and TACS, well, mostly TACS. But I think the multi-channel experimental apparatus makes an interesting playground for deep brain stimulation. Tricky to do currently with TMS, more easily done with focused ultrasound because you can actually stimulate quite deeply, but people don't understand the mechanism very well right now, and aberrations through the skull make it a little difficult with focused ultrasound. I didn't get a chance to talk about that, but that's another possibly quite interesting deep brain modulatory technique. So we'll see. I think it's not so simple as this paper makes out to be, and it may not work very well deeply. But in any case, it does allow you to shift things around in a controlled fashion, and that's pretty powerful.
Yeah, go ahead.
It's not related to that, so if you have a follow-up, that's fine. I said it wasn't related to what you were just talking about, so if you had a comment on that, go ahead.
That's okay. Yeah, go ahead.
Okay. I just wondered about the idea that fMRI is supposed to be kind of representing more the input to an area, right? So that's kind of been out there for a while, but I don't feel like it's really changed the way that people think about fMRI. Right, so then we report results and sort of this typical let's find the blob idea, right? Then they attribute sort of that action or whatever is being modulated to that region, right? And really that's implying that it's whoever is feeding into it. So I wondered sort of what you thought about, you know, how significant and how people should try to rethink how they think about those things, particularly also with resting state functional connectivity.
I actually don't think that people should change the way they think. In this morning's lecture, I did present the results by Dr. Logothetis, but I emphasized that an input to any neuron is not only input that is of origin in other areas. It's not an input to an area. It can be also input from neurons that are 50 micron away. And in fact, 80% of all the synapses that impinge on any single neuron in cerebral cortex are with origin of half a millimeter around this area. So while we try to do our best to say, you know, things like fMRI represents synaptic activity more than action potentials, fMRI represent input to an area, et cetera, et cetera. If you notice that my take-home message is that in really the majority of the studies, the real majority of studies, we need to think about fMRI as representing the overall neuronal activity within the voxel. I think that trying to do anything beyond that fits possibly 0.1% of paradigms that we look at. It's difficult to do with functional MRI, especially in the cerebral cortex, where everything is connected to everything. In every single cortical column, if we have an electrode orthogonal to the cortical surface, cortical layers are so much connected between themselves that increase in connectivity, increase in activity in any cortical layer immediately is propagating throughout the column, perhaps except for layer 6. So my recommendation is to still, with functional MRI, and especially if we talk about 3 Tesla, but even with 7 Tesla, it's very difficult to make interpretation beyond just functional MRI represent the overall activity within the voxel. That's my take-home message on that.
Okay. Yeah. Go ahead.
Yeah, I was going to ask about reproducibility and kind of a basic question, but what do you think replication is? Is our goal to replicate p-values or effect sizes, similar patterns of results, and just kind of get some feedback on that?
I don't think there's any one answer. I think p-values would be a bit dangerous, or not dangerous, significance, because you can be just above significance or just below significance. So choosing a threshold is probably not the best way to see if something replicates. But looking more at the effect estimates, the spatial locations, and I think with all the different potential combinations of ways to analyze your data. I mean, if you're talking about replication, the most stable findings should be platform independent, should be analysis independent. I mean, those, if you're looking at, for example, I think of studies that we've done, what are the effects of low folate during pregnancy and downstream effects on brain development. So you would hope that those would replicate irrespective of the different types of platforms or analysis strategies that you use. Now I'm coming, did you say reproducibility, replication? I'm sorry.
Yeah, that's good. I think it was about what should we be reproducing, right? Or replicating.
Because, yeah, replication is different. Then reproducibility comes back to really using people's own data to come up, and their analysis strategies to come up with the same results that they had. And I think showing, like the Open Science Consortium, they showed both, is it significant or not, and they also showed what are the differences in the effect estimates. And I think showing effect estimates shows you the variability of the findings.
And one idea to quickly follow up on that is we shouldn't be reproducing people's experiments exactly, but their conclusions, right? I mean, you want to be able to know that you can do something over and over again and it'll work, So I'm very utilitarian in that way. We want to know what we should believe at the end of the day is what this is all about. I have a follow-up question about bold physiology, and it has to do with, so some work by Tori Vasquez and Sanji Kim, I believe, has shown that if you optogenetically stimulate inhibitory interneurons, you also get bold increases, which is one nice demonstration. So I guess, what about high field imaging of different cortical layers? So I think some people think that might give you some leverage into saying, deciding what's a kind of inhibitory activity and what's excitatory.
Well, I would separate, or I'll allow myself to split your question into 2 parts. The first part is about optogenetic stimulation of inhibitory neurons. And that's actually, I presented a slide on that today from Anna DeVore's laboratory. And what the results from Anna DeVore show is that you get first a transient phase of increase in bold signal. But, or actually, in their case, it was the dilation or constriction of arterioles. So you get first a dilation of arterioles after optogenetic stimulation of inhibitory neurons. But then it is followed by a long and quite significant constriction. Okay? So now, no matter what, it's not news that inhibitory neurons require energy and require oxygen and glucose to function. However, one thing that we need to take into account is what is their effect, the overall effect on the network. And inhibitory neurons impinge their synapses on pyramidal cells in very strategic locations. So most of them have synapses right at the soma, or even at the soma hillock. So their effect is very strong. Although we have in cerebral cortex only about 20% of the neurons are inhibitory, they have a very strong effect on the overall activity in each location, simply because their effect or their synapses impinge on very strategic locations in terms of the production of action potentials, in terms of pyramidal cells. Another thing to consider is that, at least in my studies, I'm very careful not to mention that negative bold is equal inhibition. What I'm saying is that negative bold, in the paradigms that I used, was associated with decreases in neuronal activity, which could be also decrease in input to an area, not necessarily active inhibition within the area. It's actually very difficult, except for now with optogenetics techniques, it's very difficult to dissociate decrease in input, tonic input or excitatory input, than local inhibition. Even if you go to recording, intracellular recording from a neuron, even then it's difficult. So the only ways that I think it can be done is via a causal manipulation, such as with optogenetics. Now, the second part of your question has to do with imaging of cortical layers. I allowed myself to split between the two because I think that they are orthogonal. The reason is that in every cortical layers, you have both pyramidal cells or excitatory cells, and you have also inhibitory neurons. So these are two different questions. Now, in my view, imaging cortical layers is difficult. It's much more difficult than imaging cortical columns. I strongly believe that functional MRI at 7 Tesla and higher, and especially with a recent method of improved RF, and like we are at the level of imaging cortical columns. There are papers going back to the beginning of the 2000, and then throughout the 2000 until now, of imaging cortical columns with very high reliability. I believe I see Dr. Menon here. He published a couple of papers on that. And so I believe that functional MRI at 7 Tesla and higher is capable of imaging cortical columns. Definitely cortical columns whose cortical cycle is 2 millimeter, about 2 millimeter, like ocular dominance columns. So I'm involved in imaging cortical columns, very highly reproducible, fit exactly what we know about the monkey visual cortex and how they are organized. So I'm a very strong believer in that, except for maybe orientation columns that are harder to get. Any other columns in the visual cortex or somatosensory cortex that we know of from the monkey model have lower spatial or a longer spatial cycle, lower spatial frequency on cortex, which means that, in my view, functional MRI at 7 Tesla and higher is capable of imaging now the majority of columnar organizations in the cerebral cortex. In contrast, I believe, well, I'm making efforts myself, and I strongly believe that we should continue making efforts because it's a very important question, but I believe that lamina-specific imaging is difficult. There are several reasons why it is. One of the main ones is that it's difficult to find paradigms that actually get you responses in specific cortical layers. And if you think you have a paradigm like that, as I mentioned a few minutes ago, cortical layers are interconnected very much. If one layer increases its activity, it immediately propagates everywhere within the same column. So it is difficult to image cortical layers. Another reason is that a part of the veins of the venules from which we get our data are organized in a manner which is orthogonal to the cortical manifold, which means that they actually smooth any activity across cortical layers. So that's another difficulty, and I'll stop here. So I think that we are at an era in which it's difficult, but it is doable to image cortical columns. It still remains difficult and challenging to image cortical layers functionally.
I'm sure that many of us would like to just press on through lunch, but I think we should give our speakers a rest. And so we're going to pause for lunch and regroup at 1 o'clock sharp. Thank you.